a quirk of public health policy in Wales, has produced one of the most striking clues in the fight against dementia. When the country launched its shingles vaccination program on the 1st of September 2013, eligibility hinged on a single, almost absurd detail: your exact date of birth. People who were 79 on that date could receive the vaccine for 1 year. People who were 80 or older on that date were permanently excluded. No exceptions, no appeals. That razor-thin age gap created something scientists rarely get outside a carefully controlled laboratory: a natural experiment with near-random assignment and minimal bias. Those who were eligible for the shingles vaccine were roughly 20% less likely to receive a new dementia diagnosis over the following 7 years. The finding was not subtle or ambiguous. It appeared across both sexes, across dementia subtypes, and it held up under rigorous statistical scrutiny. This eliminated the so-called healthy vaccinee bias, the well-known problem that people who seek out vaccines tend to be healthier in the first place. By comparing people separated by mere days of birth, the study sidestepped that bias entirely. Nearly everyone alive today carries a silent passenger. The varicella-zoster virus, responsible for childhood chickenpox, never actually leaves the body. After the rash fades, the virus retreats deep into nerve cells and enters a dormant state that can persist for decades. In roughly 1 out of every 3 people, it eventually reactivates, emerging as shingles, a condition notorious for its excruciating, blistering rash. But the virus does not need to produce visible symptoms to cause damage. Even in its dormant phase, it drives chronic inflammation in the nervous system that quietly accumulates over a lifetime. When neurons harboring dormant herpes simplex virus type 1 are exposed to the varicella-zoster virus, something dramatic happens. The varicella-zoster virus triggers inflammatory signals that reawaken the sleeping herpes simplex virus. That reactivation unleashes a cascade of Alzheimer hallmarks, surging levels of tau protein and amyloid beta plaques, along with a measurable slowdown in neural signaling. The varicella-zoster virus alone does not produce those signature proteins. Instead, it acts as an inflammatory match, lighting a fuse already embedded in the brain. Herpes simplex virus type 1 infects roughly 90% of the global population, and varicella-zoster infects about 95% of people before age 20. This 1 to 2 viral punch represents an enormously common risk pathway that science is only now beginning to map. Shingles vaccination does not merely reduce the odds of developing dementia. It appears to actively slow the disease in people who already have it. Those who received the shingles vaccine saw a 29.5 percentage point reduction in their risk of dying from dementia over a 9-year period. The vaccine also reduced new diagnoses of mild cognitive impairment, the early warning stage that frequently precedes full dementia. These benefits appeared across the entire disease course, stretching from the earliest detectable cognitive changes all the way to the most advanced stages of decline. Data from more than 174,000 adults across 107 U.S. health systems revealed even more. Vaccinated individuals showed a 50% lower risk of vascular dementia, a 25% lower risk of heart attack or stroke, a 27% lower risk of blood clots, and a 21% lower risk of death from any cause. The shingles vaccine may be protecting both the heart and the brain through the same anti-inflammatory mechanism, quieting a virus whose repeated reactivations erode vascular and neural health simultaneously. Scientists compared the shingles vaccine Shingrix with the RSV vaccine RXV. These two vaccines target completely different viruses, but share one key ingredient: the AS1 adjuvant system, an immune-boosting compound. Among more than 436,000 adults, both vaccines independently lowered dementia risk within 18 months. Shingrix recipients showed an 18% reduction. RXV recipients showed a 29% reduction, and those who received both saw a 37% lower risk. No significant difference existed between the two vaccines in their dementia protective effect, despite targeting unrelated diseases. This strongly suggests the AS1 adjuvant itself is driving the brain benefit, not merely the prevention of shingles or RSV infection. The adjuvant contains two potent immune stimulators, monophospholipid A, known as MPL, and a plant-derived compound called QS21. Animal studies have shown that MPL stimulation improves Alzheimer-related pathology in mice, likely by activating immune cells called microglia that clear harmful proteins from the brain. The protection appeared within just months of vaccination, far too quickly to be explained by preventing enough infections to meaningfully reduce cumulative viral damage. Something in the adjuvant appears to directly recalibrate the immune system's relationship with the aging brain. Dementia currently affects more than 55 million people worldwide, with roughly 10 million new cases diagnosed each year. Projections suggest the number could reach 139 million by 2050. Despite decades of research focused on amyloid plaques and tau tangles, no breakthrough in prevention has emerged from that approach alone. The growing evidence around shingles vaccination represents a fundamentally different avenue, one rooted in the interplay between chronic viral infection, immune system aging, and neuroinflammation. It suggests that dementia may be, at least in part, an immune disorder of the brain rather than purely a disease of protein accumulation. The shingles vaccine is not some theoretical compound awaiting years of development and regulatory approval. It already sits on pharmacy shelves worldwide, recommended for adults 50 and older. Clinical trials are now being planned to formally test whether the vaccine can serve as a dementia intervention. The shingles vaccine has been identified as the single most promising existing medicine that could be repurposed for Alzheimer prevention or treatment. It requires only 2 doses and carries a well-established safety profile. A vaccine designed to prevent a painful rash might simultaneously protect and even treat the most feared disease of aging. This is a hypothesis backed by natural experiments, massive data sets, laboratory models, and a growing understanding that the viruses sleeping in our nerves have been quietly reshaping our brains all along. As our journey through understanding complex topics comes to an end, we hope you've gained some valuable tools and perspectives from this exploration of diverse subjects and tasks. The research highlighted, like the surprising connection between shingles vaccination and reduced dementia risk, a 20% reduction revealed by a unique Welsh public health policy, demonstrates how unexpected discoveries can emerge when we look at things differently. Remember that even seemingly unrelated areas of study, such as viral infections and neurological diseases, can hold profound insights for one another. Thank you for watching. Please leave a like and share your own experiences or hidden gems in the comments below.